Hello everyone, today we are going to dive into the essential topic of growth monitoring in microbiology. As you can see, this is a crucial practice for ensuring quality, safety and efficiency across many fields, from food and pharma to diagnostics. The constant challenge we face is that microbes have extremely fast generation times, which means we need frequent and rapid monitoring method to keep up. On this slide, we are highlighting why this is so crucial. In fermentation industries, for example, we monitor growth to ensure we get peak production of valuable products like antibiotics, enzymes, or even ethanol. In clinical settings and diagnostics, monitoring helps us determine the effectiveness of antimicrobials, a practice known as measuring the minimum inhibitory concentrations or MIC. And in basic research, it's how we study everything from how fast microbes grows to what its metabolic pathway are. Now let us look at some of the traditional methods for directly counting microbes. Our first method is hemocytometry or direct microscopic count. This uses a specialized slide with a grid. You place a known volume of your sample on it, count the cells, and then use a simple calculation to find the total cells per milliliter. It's fast and gives you a total cell count, but it can be tedious, and without special stain, you can't tell the difference between living and dead cells. Next, we have plate counts, also known as the viable cell counts or colony forming units. This is a classic method that measures only the living viable cells. You take a serially diluted sample and either spread it on the surface of an agar plate or mix it in with the molten agar. After incubating for 24 to 72 hours, you count the colonies that have formed. The big advantage here is that it only measures living cells, but as you can imagine, it's very time consuming. Moving on to indirect method, which measures the property of culture that's proportional to the cell number or mass. Turgidity or optional density is a great example. This method uses a spectrometry to measure the amount of light scattered by the suspended cells. It's an instantaneous method that is non-destructive, and it can even be used for continuous monitoring. However, a major drawback is that it can't distinguish between living and dead cells, and it can become unreliable in very dense cultures. Another traditional indirect method is dry weight or wet weight. Here, we harvest the cells from the culture, wash them and dry them to a constant weight. This is the most accurate measure of total biomass, but it's extremely slow and destructive, making it unsuitable for real-time monitoring. We can also measure a specific cell component like DNA or protein, but these methods are also indirect and can be affected by the cell's physiological state. Now this brings us to advanced and automated techniques that provide faster, higher resolution data. Automated turbidity systems with inline sensors in bioreactors allows for continuous real-time growth curves. Flow cytometry is a high-resolution method that uses lasers to rapidly count and analyze thousands of individual cells per second. And automated colony counters uses digital image processing to quickly and accurately count colonies on multiple plates, saving a lot of manual labor. Beyond just counting cells, we can monitor a cell's health by looking at its metabolic indicators. We can track substrate consumptions to see if the cells are actively using their food source, or measure the fermentation of the products like ethanol or desired protein. Monitoring dissolved gases such as oxygen and carbon dioxide gives us real-time data on the culture's overall respiratory activity and metabolic state. It's important to remember that no single method is perfect for every situation. The choice of technique depends on your specific goal. Do you need total cells or viable cells? How quickly do you need the results? What kind of microbe are you working with? Often the most complete picture comes from using a combination of methods. Let's look at a few examples of where these techniques are applied. In a clinical lab, rapid blood culture system using impedance can quickly diagnose sepsis. The food industry uses ATP bioluminescence for rapid hygiene checks on surface. In a research lab, we often use spectrometry to quickly monitor growth. And in biotechnology, sophisticated bioreactors monitoring helps optimizing the yield of valuable products. In conclusion, microbial growth monitoring requires a balance between absolute viable counts like CFU and rapid indirect methods like optical density. Modern biotechnology is driving the need for non-invasive high-frequency data. The future will involve integration, multiply inline sensors, and using machine learning to fuse all this data together to predict accurate cell count in real time. Ultimately, continuous and rapid microbial growth monitoring is crucial for robust, reproducible, and compliant process in industrial microbiology. Thanks for watching.